Hello friends, we are studying about the TYBSc Botany paper, paper 3 in semester 3rd, that is BO 333 Genetics and Evolution. In this we are studying the second chapter, Mendelism. Okay, in this chapter we have this content which we are going to study. All these terminologies, different laws that were proposed by Mendel, and then back cross and test cross. I am Dr. Sangeeta Kulkarni from the Department of Botany, Radhabai Kale, Mahila Mahavidyalaya, Ahmednagar. So in this topic, we are going to study what Mendel had worked or what was Mendel's work. So in the previous chapter, I had told you that Mendel, he was called as father of genetics, and he has worked on the pea plant, that is Pisum sativum. Mendel, he had selected this pea plant, and he was very lucky that after the selection of this plant, he had selected 7 contrasting characters which were present in this plant for study. Okay, naturally this plant is a self-pollinating plant, but cross-pollination, he had carried out studies using cross-pollination, and he got the perfect results for that. So there are various benefits of using this plant, and therefore he selected garden pea for his experiments. Which were the characteristics that we will study. The flowers are bisexual and naturally self-pollinating. They are artificially cross-pollinated because of the experiments he had carried out using both. Naturally self-pollination was there, but he also did cross-pollination between plants to get the perfect results. The offspring produced after cross-pollinations were found to be fertile. In most of the cases, if it is a self-pollinating plant, there are chances that it will not cross-pollinate or become fertile. But in this case, it was fertile. Pea plants have several pairs of contrasting characters. Flowers of pea plant are large enough for easy emasculation, that is for cross pollination. They are very big flower, it can be opened and cross pollination can be done. And it is a small herbaceous plant, so he could grow large number of plants. So because of all these characters, Mendel, he was successful in his experiments, and luckily all the 7 characters which he had studied, shape of the seed, color of the seed, color of the pod, shape of the pod, then the plant height, position of the flower and the flower color. These were 7 characters which he studied in 2 forms. One was dominant, other was recessive, and whatever he had studied were luckily present on 7 different chromosomes which were assorted independently, and therefore he got the results according to his choice. Okay. Now the first cross which he conducted was monohybrid cross. What is a monohybrid cross? In an experiment, when only one trait or character is considered for crossing to create hybrids, it is called as a monohybrid cross. Mono means single. So when we use only in any experiment to test a character or to study a particular character, we use only one character at a time. It is called as a monohybrid cross. Okay, and the ratio obtained after this cross in F2 generation. F2 means second filial generation. F1 means first filial generation. So ratio which is obtained after cross in the F2 generation, that is after one generation, it is termed as a monohybrid ratio. That means in the next generation, when you get whatever ratio you get, it is called as a monohybrid ratio. The phenotypic ratio, monohybrid ratio, is 3:1. That means totally you get 4 individuals, out of which 3 are dominant and 1 is recessive. Okay, so this you are able to study after this monohybrid cross. Okay, now how this monohybrid cross is done? The parents are selected of homozygous parent. Homozygous means having identical pairs of alleles. So this is supposed to be the genotype, capital T, capital T. We have taken this as a genotype, capital T, capital T. Another is crossed with small t, small t. Okay, that is homozygous recessive. This is homozygous dominant. Okay, so all the gametes of this will be of same type. Therefore, we have considered 1T. Gamete means only one at a time gamete we have to consider, and here we are considering small t. So when we cross this capital T and small t, it becomes capital T and small t. This is a heterozygous tall. Why it is tall? because it was found that one which is dominant character is always expressing. So here capital T is expressing, therefore we say that this is tall. Again in the next generation, when we have to segregate or separate the gametes, this is of two types, so automatically the gametes will be of two types. Okay, capital T and small t. Here why we have taken 1? Because both are same, therefore 1. So this is a homozygous condition. This is a heterozygous condition. Okay, for heterozygous we have to divide it into number of whatever is the total number of phenotype. Based on that we are going to separate them as capital T and small t. We are segregating the alleles into two types so that in future they can be used for further crossing. Okay, this is a monohybrid cross. So for monohybrid cross you have to make a cross like this, capital T crossed with small t. The gametes for capital T is T and gametes for small t is small t is T. So after combination in first F1 generation we get capital T small t heterozygous condition, which are all tall. Why? Because the tall condition is dominant over the dwarf condition. Okay. Now in the F2 generation, after this to get the monohybrid ratio, this cross was continued. So now in the first part we had studied this, this was the cross pollination that was done between two plants, and in the second part cell pollination was done. Okay, this is a plant containing this genotype, and another it was selfing was done, that is in the same flower there was combination was done. So it was producing two types of gametes, this was also producing two types of gametes. Now Mendel, he made a Punnett square. Okay, he did the calculation in the form of this square called as a Punnett square. So he arranged the male gametes on one side and the female gametes on one side, and these are the possible, you can say, combinations. Capital T because both T are joined, then here capital T small t, here capital T small t, and here small t. Therefore this is the phenotypic ratio, that is total population 75% are tall and 25% are dwarf. Okay, so this character, even if it is recessive, it is expressed in the second generation, not in the first generation. Therefore you find 3 tall and 1 dwarf. 3 tall means out of the 100% population, 75 will be tall and 25 will be dwarf. But if you study it genotypically means the genetic makeup, if you study, then 1 will be capital T, 2 will be capital T small t, and 1 will be small t. But phenotypically we say that the ratio is 3:1 because out of this these will be tall, expressed tall, and this will be dwarf. Okay, so this is monohybrid ratio that is seen after you cross a single trait. Anything single, when you cross you get a ratio like this in phenotypic ratio. Okay, based on that Mendel, he proposed the law of dominance. When in a monohybrid cross, two homozygous plants with contrasting characters are crossed, in F1 only one character appears, as you have seen that capital T small t has appeared. The characters that appear in F1 offspring are called as dominant characters, and the characters which do not appear or suppressed, they are called as recessive characters. This is law of dominance. When a monohybrid cross is conducted, two homozygous plants with contrasting characters are crossed. In F1, only one of them is expressed or appears, it is called as dominant, and the other one does not appear or remains suppressed, called as recessive character. Example, when a cross is made between a homozygous tall and a dwarf plant, the first generation offspring were heterozygous, capital T small t. That means that tallness character was dominant over dwarfness characters. Okay, this was tall. Why this tall has come? It is because T, capital T is dominant over small t. Okay, now next, after the study of law of dominance, various people, they have studied the same effect, or they wanted to study the same effect in different plants. But when they carried out the experiments in plants, some of the plants they followed the law of dominance, but not all the plants they followed. There were exceptions to that. Okay, so based on that, even new concepts, or you can say new laws, were formulated. One of them was incomplete dominance. Okay, So in this case, what happens when a dominant allele does not completely mask the effect of recessive allele and the resulting phenotype shows blending of both, it is called as incomplete dominance. Okay. So when what happens, there is a dominant allele, there is a recessive allele, but none of them will express, neither this is dominant completely, now neither this is recessive completely. What they are doing, they are blending together to show an effect of both the alleles. So here in this case you can see that a red flower is crossed with a white flower and they produce three types of flowers now. Instead of only red, or you can say only white, they are producing pink type also. That means what has happened, there is blending of red and white color to produce a new type of color that is called as pink color. This is another, you can see that there is a dark purple and there is a white colored parents, and when they are crossed you get this type of offspring. Okay, so you are having three types of flowers. Now why? Because of none of them are dominant, considered dominant. Therefore this is called as incomplete dominance. So this was studied in flowers like roses, tulips, carnation, snapdragon. These are some of the examples which are showing incomplete dominance. I told you that all the plants do not show complete dominance, but some of them may be showing this type of dominance called as incomplete dominance. It is different from the codominance. There is one more term in dominance called as codominance, where both of them express together. Okay, and therefore when they express, the expression is something different. Okay, they do not blend with each other, both of them express same. Okay, it is called as codominance, that we are going to study further. Next concept is codominance. There is one more exception to law of dominance, that is called as codominance. Okay, this is one more effect that is seen. Now here I have taken these pictures from Google to show you that what is the concept. Therefore these pictures are taken. So this is owl and this is flower. Both these are same, only I will try to tell you that how this codominance works. See, this is capital R, which is showing the reddish or brownish color in owl, and this is a grey colored owl showing the genotype capital G. But in case of law of dominance, either of them should have been expressed, but this does not happen. Or in case of incomplete dominance, there should be blending and showing a resultant color which is different from other, these two should be brown in color. Okay, but this has not occurred. Instead you can see patches of this reddish color as well as patches of grey color. What has happened here, that both the genes, they have expressed together, and because of which half part has become red and half this one red face, and the remaining part has become grey in color. Okay, so the picture itself depicts that what is the combination, that neither is dominant, neither is recessive, neither is incomplete, it is codominance. Both are working together without blending with each other. In the previous one what we had studied, that blending creates a new color. Here we don't create a new color. Here co means together. So here both the alleles show dominance when together without blending. Codominance occurs when two alleles in heterozygous condition are fully expressed. Neither allele can dominate, so they do not blend. This is another example of codominance seen in camellia flowers. If you can see the red flower is crossed with a white flower, the alleles are codominant, therefore you get the mixture of both these colors. Okay, you don't get a pink color flower, but instead you get patches of red and white together, and therefore this is called as codominance. These are exception to the law of dominance. Therefore I have shown you here that how these structures or these characters, they are inherited from parents to the offspring. Now the second law was proposed by Mendel, was called as law of segregation or law of purity of gametes. The law of segregation is in a monohybrid cross. When two homozygous parents are crossed for a single character, their alleles come together in F1 generation. When the gametes are formed in F1 individuals, these alleles segregate or separate from each other and enter in different gametes. They are pure for their characters. Okay, so here what has happened, when we have studied this in monohybrid cross, capital T capital crossed with small t, F1 generation shows capital T small t, which is a heterozygous tall. And what we are saying, that in next generation, when at the time of gamete formation, these two structures will separate, they are pure. Capital T will always show tallness and small t will always show dwarfness. Okay, and therefore they will be separated from each other and they will be pure for their characters. This is the law of segregation. Segregation means they will segregate or separate at the time of gamete formation and they are pure for their characters. This is called as law of segregation. Okay, now the next cross. After the first success of monohybrid cross, Mendel, he carried out the dihybrid cross. Okay, so he used two characters at a time. Instead of one character, now there were two characters at a time for study, and automatically the ratio was also different and he could find much variation in that. So what is a dihybrid cross? It is a cross between two parents that differ by two pairs of alleles. Okay, it is a cross between two parents that differ by two pairs of alleles, or when two pairs of characters are considered for crossing, it is called as a dihybrid cross. Instead of one character, we are using two characters. So example is capital A is one character, capital A is one gene, and capital B is another gene. So we are considering two genes at a time. Okay, the exactly opposite of it is small a and small b. Here this is one gene, this is another gene. Here also for this this is the contrast. Okay, for everything there is a contrast. If one is dark, another is light. This is yellow, this is green. This is tall, this is dwarf. So we consider two parts of the same gene. So this is capital B, opposite to it is small b. The phenotypic ratio obtained in F2 generation by such type of cross is called as a dihybrid ratio. We had studied the monohybrid ratio, that in F2 generation we get a phenotypic ratio of 3:1. Here the phenotypic ratio in F2 generation after the cross is called as the dihybrid ratio, which is 9:3:3:1. Okay, totally we get 16 individual. Out of what the result is in this manner, 9:3:3:1. Okay, so this is what happens in the dihybrid cross. Now again same way we are studying the crossing over between the two characters that we are studying. So parent are yellow and round seeds, and another opposite to that are green and wrinkled seeds. So yellow is having another form green and round is having wrinkled. Okay, so one character which is considered for study is yellow round and the other plant was taken as green wrinkled. So the genotype for this is capital Y. So wherever you get this capital YY or single Y, they are always going to show you yellow color. Okay, and capital R means this is round. Even single or double E, they are going to express as round. Okay, small y when they come together, they will express green color, and small r they are considered as wrinkled. Okay, so the gametes for this singly, for 2y it is a single y, for 2r it is a single r. So yr is the gamete. Here small y small r is the gamete. When these two combine, they are going to produce a heterozygous yellow round. Why yellow round? Because capital Y is present, which is dominant over small y, and capital R is present, which is dominant over small r. Okay, so this is what happens. Selfing takes place, and in F2 generation, instead of now 2, you get 4 gametes. 4 gametes you are getting. There you were getting capital T and small t. Here you are getting 4 male gametes and 4 female gametes. Now based on this, the dihybrid cross is studied. This is how the dihybrid cross is studied. You are able to see that on one side of the Punnett square we have written the male gametes, on the other side we have written the female gamete. Then we have made the combinations. In this combination on the board you can see that 9 are yellow round YR, which are containing capital Y and capital R. You can see this is one, this is one capital Y, this is another capital Y, capital R, capital Y, capital R, capital Y, capital R, capital Y, capital R, capital Y, capital R. Not purposely I have kept only these blue ones. Okay, this is capital Y, capital R, capital Y, capital R, and capital Y, capital R. So this is 4, 5, 6, 7, 8 and 9. These are yellow round because they are containing the dominant gene yellow and round. This is yellow and wrinkled because smaller is present. So how many of these are present? Yellow and wrinkled. This is one YR, Y and small r. This is another capital Y small r, and this is third one capital Y and small r. Okay, so these three are considered as yellow and wrinkled. Then this one is green. Y is small, so it is green, and capital R is present, so round. So one is this round, green round. This is green round. This is green and round. Okay, so three are green and round, small y and capital R. And this last one is homozygous recessive, green wrinkled. Okay, so one is a homozygous dominant, capital Y, capital Y, capital R, capital R. The last one is small y, small r. This is homozygous recessive, and remaining 14 are hybrids. But these hybrids are showing yellow round structure. Okay, so this variation is seen, and the ratio you are getting 9:3:3:1 in a dihybrid cross. So you have to prepare a dihybrid ratio in this manner, and you are going to prove that the result is 9:3:3:1. Based on that, Mendel, he has given the law of independent assortment, which states that when you are using different gametes in a dihybrid cross, all the gametes, they segregate at the time of gamete formation and are assorted independently. That is, their behavior is independent to each other, and therefore you are able to get all these combinations. The definition is already given here, that they assort independently and they give their expression independently. Therefore you are getting yellow separate, you are getting round separate, you are getting wrinkled separate, and you are getting green separate. Otherwise, if they would have been linked, you would not have got this result. Okay, so this is the law of independent assortment. Capital A, small a, capital B, small b are segregating separately, and they are independent to each other. Now next part we are going to study is back cross and test cross. Okay, what is back cross? After the study of all these crosses, the next cross that was conducted, when F1 individuals are crossed with one of the parents they have been, from which they have derived, it is called as back cross. This cross is done, basically it is done to show or to improve the characters of the offspring. So every time it is back crossed with one of the parents, basically to show that back cross, it helps in improving the characters of the offspring. And another one was test cross, which was basically conducted for a heterozygous with a recessive parent to show whether this genotype is heterozygous or homozygous. For this was conducted with the one of the parent. This was either of the parents. So test cross is one of the back crosses only. So this you can see how back crossing is done. It is useful in genetic studies for isolating certain characteristics in related groups of plants and animals. So A is crossed with R, B is again crossed with R, C is crossed with R, D is crossed again with R. So every time you are crossing it with one of the parents so that you improve the characters. Okay, so this is the main aim of back crossing, which is done and to improve the characters. So you increase the hybrid vigor. So in case of plants, this is usually done. Now test cross, you can see that when it is crossed with the recessive parent, you get this type of combinations in monohybrid cross and this type of combination in dihybrid cross. So these are taken pictures from the Google, and these are basically to show that whether the conditions are homozygous or whether they are heterozygous, you test it, and based on that you are able to study it. So these are important crosses which were conducted by Mendel and proved that they are responsible for showing the characteristics of the population. Now what are the uses of test cross? Test cross are used to test the individual genotype by crossing it with the known genotype. Okay, individuals that show recessive phenotype are known to have homozygous recessive genotype, while those having heterozygous condition are showing heterozygous recessive genotype. So you come to know whether it is dominant or whether it is recessive based on that. Okay, thank you very much for completing this second chapter.